Welcome to Molecular Nutrition, where food becomes molecules, molecules power cells, and cells sustain life. So if you recall from the previous lecture, we established that membranes are selectively permeable, so which means that as barriers, acting as barriers to the different molecules and substances crossing them, they allow some molecules or some substances to pass while preventing others from passing. So meanwhile, in between, others will require some form of assistance. So these three concepts are very important when we are thinking about how different molecules pass. And we can then look at those substances or molecules that we're talking about. So some of them are ions, and we know by nature ions are charged. And we can see, for example, here sodium ion, a very charged ion. So if you remember the atomic mass, atomic numbers of sodium, and the difference between an atom and an ion in terms of loss of electron. So sodium ion here is charged, and because it's charged, it's going to be very difficult for it to pass through the phosphate barriers of the membrane. So we move to other substances like sucrose. The same for sucrose. If we look at sucrose, it's a disaccharide with two glucose molecules. It's also difficult for sucrose to pass because of its size. So if we look at the molecular mass of sucrose, I mean it's quite heavy. C2H22O11. It's heavy, and as a result, it's difficult for it to pass. However, we have non-polar molecules like gases. So if we think about how do the gases pass, of course they're so small that they can pass through the membrane. This is nature's wisdom. Imagine if your gases cannot pass through the membrane. You're going to be easily bloated and you will burst like a balloon because we're constantly generating a lot of gases. Okay, so however, we can also consider small polar molecules. So although polar, like water, they are small, right? So they are also able to pass, but very slowly. So we'll be coming to that in the next couple of slides, where we'll see that water passes through the membrane, but it passes very slowly. Now, if we think about other polar compounds like ethanol, also because of its small size, it's also able to pass the membrane. So in a nutshell, we can see as a result of the structure of the membrane, we can see how it selectively allows certain compounds to pass while preventing others to pass, while others will require assistance. And this is what we call the selective permeability of membranes. In this slide, we see specifically, so in the same idea that we have been communicating, we can see what are those physical structures that make the membrane selectively permeable. So we already established that. We already established in the previous slides the different components of the membrane. So first we can see a hydrophilic head. And what is this hydrophilic head? The hydrophilic head is made up of the phosphate group. So if you look at the head of the membrane, it's actually made up of phosphate, and it has phosphate bound with oxygen, and this is what makes up the hydrophilic head, and the same for the hydrophilic tail. So earlier on you saw the video that we considered looking at oil and how oil behaves with regards to water. So it's the same thing. And in between we have the fatty acid tail. And the fatty acid tail is water repelling. It's hydrophobic. And that's why it's difficult for oil and water to mix. And it's the same concept of what makes the membrane impermeable to certain compounds. Now, in aqueous solution in the cell, you find that the phosphate group, like we saw in the oil and water video, aligns to the water area in the extracellular side of the membrane and also in the intracellular side or cytoplasmic side of the membrane. So we already understand this. So this creates a hydrophobic interior layer, and it therefore makes this membrane, the membrane, impermeable to water or any polar compound. So we already established how small non-polar molecules will pass easily and how large polar molecules wouldn't be able to pass, while at the same time ions that are really charged will not be able to pass at all. So it's quite important for you to get a hang of it, so to understand what kind of compounds do we usually find in the cells, and which ones will need special channels, special transporters to pass, and which ones can really diffuse easily across the membrane. So with practice, you will get a hang of that. Okay, so, but one thing that can help us in our thinking, so maybe I tell you, okay, so oxygen is not here, but imagine you have oxygen. Will oxygen be able to pass? Yes, oxygen is a gas. So will chlorine or chloride ion, so let's think about chloride ion, if we replace this sodium ion with chloride ion, will it be able to pass? No, because chloride ion is highly charged. So we can look at those factors that that affect permeability, so that we can apply them as a general rule. So the first is the size of the molecule. If the molecule is large, like sucrose, like glucose, like peptides, it can't pass through. If it's polar, and what do we mean by polar? So if you look at water, you remember in secondary school we used to ask a common question, that what is the difference between polarity and charge? So what is the difference? If we look at sodium ion here, sodium ion here is charged because it has lost one electron and it's an ion, it's heavily charged. But polarity is also a kind of charge, but it has to do with distribution and overall charge. So if we look at water, we see that water has a kind of a net charge. It's not charged, but it's polar because it has a delta negative and a delta positive. So overall, it's somewhat charged. And we can look at different compounds in that sense, but polarity and charge are different. So going back, these are factors that can affect how a compound or a substance crosses or does not cross the membrane. So general principle, if it's polar, it's heavily polar, it may not be able to pass. If it's slightly polar like water, it will pass very slowly. If it's charged, no way, it's a no-no. So any ion cannot pass through the membrane. For an ion to pass through the membrane, you will need specific ion channels, right? And we talked about that earlier on. And if it's lipid, so if it's soluble in a lipid, yes, it will diffuse through the membrane, aided by the lipid tails and the lipid layer of the membrane. So these are the general principles: the size of the molecule, the polarity, the charge, and lipid solubility. So some other concepts to consider is the way the lipids are tightly packed to prevent large molecules from passing, and that's why only small molecules like gases can and water, but very slowly, can pass through, and they can slip in between the membrane. Small molecules can diffuse through the membrane, like I said, but because they can fit easily in between the membranes and they can move via diffusion. So capping it up, we then talk about the fluid mosaic model of the membrane. And it's there are different models of the membrane, but this is one of the most popular and modern model of the membrane. So having done an established study, and a very thorough study, extended study, we then over the years have accepted as the closest to reality and to actually what happens in the cell, the fluid mosaic model that describes the membrane. So when you think about the fluid mosaic model of the membrane, what should come to your mind? It's everything that we have discussed so far. And the first thing is the lipid bilayer. As we have seen, that some proteins are embedded, and they transcend the double phospholipid bilayer, but some do not, and the ones that do not are called the peripheral proteins. The ones that transcend the double layer, they are called integral proteins. And the barrier is phospholipid. This is part of the fluid mosaic model, as we have discussed, the phosphate heads, and they are hydrophilic. And we have the lipid or fatty acid tail, and they are hydrophobic, water hating, hydrophilic, water loving. Philos in Greek is love, hydro, water, philos, water loving. Okay, so, and if you want me to apply the same logic, hydro, water, phobia, hatred for water. So hydrophobic is like hydrophobia. Okay, so and lastly, we also have, as part of the fluid mosaic model, the cholesterol in between the fatty acid layer of the membrane. We see the cholesterol, and it plays a huge role in membrane fluidity, and as we did that and established that earlier on. Okay, so in this slide we're going to be looking at transport across biological membranes. So how do substances, how do they transport? To make it easy for us to understand the transport, we're going to be looking at the transport systems as under three generic arms. And the first is the passive transport. We're going to be looking at what entails the passive transport, the active transport, and vesicular transport. So again, try to memorize, first of all, passive transport, active transport, and vesicular transport. So we're going to be looking at them one by one, and we're going to be looking at examples of the different processes, the different mechanisms in the membranes for the different categories of transportation. So, considering passive transport, what are the general characteristics of passive transport? A very key characteristic is that it doesn't require energy. And this is very important in the cell, because energy is something that the body needs to conserve, and the body is a master conservator of energy. And there is a kind of wisdom to this, because if the mechanisms of the entire systemic mechanism of the body is such that it doesn't conserve energy, then it's really going to be a lot of burden on us. So either we're going to be really needing a lot of food, or it's not going to be efficient. So one of the mechanisms in which the body conserves energy is, so if I need to move molecules around, then if I don't need energy, then I don't need to use energy. And this is where passive transport happens. There are certain molecules that, yes, really, we don't need energy to move them around, so they diffuse easily. And that's why under passive transportation, we have diffusion, simple diffusion, we have facilitated diffusion, and we have osmosis. So from your secondary school biology, you remember what simple diffusion is, you remember what osmosis is. So those are prerequisite knowledge, but we'll be reminding also in a bit. Okay, so it's then good to understand, like, substances like gases, like nitrogen, oxygen. So think about, how does your oxygen in your lungs, how does it enter the alveoli? Obviously it goes in through the epithelial cells, and the membranes in the epithelial cells, they are such that the oxygen just passively diffuses without any hindrance. It just diffuses. Okay, so we know that diffusion, when we're thinking diffusion, we're thinking about the movement of molecules from a region of higher concentration to the region of lower concentration, in this case, through the lipid membrane. We already know that. And like I said, gases belong to this category. The same thing, non-polar substances or slightly polar substances, like we already established, like fatty acids, water, slowly, right? And glycerol and steroid hormones, most of them diffuse passively through the cell membrane. So one of the key characteristics of passive diffusion is that you don't need energy, like we have already established, and you don't need those membrane proteins. A movement just happens down the concentration gradient from a region of higher to a region of lower concentration. Okay, so I already talked about how oxygen moves across the lungs in the alveoli. So this is an example of passive diffusion. Now, if we think about facilitated diffusion, it's quite different. And in this case, molecules are moving from a region of higher concentration to low concentration across the membrane, but this time with the help of membrane proteins. So you look at previous videos where you see embedded in the membrane are proteins, which we already discussed. So those proteins aid in facilitating the movement of certain molecules across the membrane, but they do so in a manner that does not require energy. And what do we mean by energy? It doesn't require ATP. Normally, the adenosine triphosphate, you will recall, ATP is the energy currency of the cell. So every transaction that requires ATP will require adenosine triphosphate. So you can think about adenosine triphosphate as a molecule with 3 phosphate groups, right? And every time energy is required, there is a hydrolysis reaction that releases a phosphate group, and that phosphate group is what participates, we'll see it later, in the energy transaction or energy reaction. So in this case, when we're thinking about facilitated diffusion, energy is not needed, but we need proteins in between the proteins that embed in the cell membrane. Okay, so there are many important molecules that really they cannot pass through the bilayer because they are charged, they are large, like we've seen earlier on, they are polar, and they will need those proteins. So all the integral proteins that we saw earlier on, this is actually where they come in. The same for water. So water, when it's needed in very large quantities, is slightly polar, and the rate with which it diffuses through the membrane is quite slow. So it will require even special proteins that will allow the water molecules to move in a very steady and fast manner. We call them aquaporins. We'll be seeing them later on. But all these are examples of facilitated diffusion. To recap, think of facilitated diffusion as molecules moving across the membrane, but with the help of special transport proteins, but still does not require energy. Okay, now it's good to look at different nutrients, so we can start thinking about different nutrients, talking about their transport strategies, if we're going to call it like that. So transport strategies, well, I call it transport strategies, but basically, it's basically how the transportation of these molecules move across the membrane. So we're thinking about the nutrients. So I just want you to conceptually think, if it's a carbohydrate that is being transported, think about it. Can it go on its own? No, it requires transport proteins. And when we think of carbohydrates, let's think about it. It's not like you're going to have your starch trying to cross the membrane. No, your starch would have been broken into glucose. So the glucose is a carbohydrate, fructose is a carbohydrate, galactose is a carbohydrate. So but they are monosaccharides. But the carbohydrates, under the general, we can still understand them as carbohydrates nonetheless, although they are monosaccharides. Okay, so they can't cross the membrane, and they usually will require transport proteins. So think about other nutrients like amino acids, because they are polar. They also require specific transport proteins. If you think about lipids are non-polar, so they diffuse easily through the membrane. And think about minerals, like we've established for sodium, potassium, chloride, because they are highly charged. They will also require either active, requiring energy, or facilitated transport that it does not require energy. But nonetheless, they also will move through transport proteins. Okay, so I've prepared a list of different compounds or molecules, and so we could do a short classwork here, and we could say, are these permeable or not? Then later on you could answer why. So first of all, think about these compounds: oxygen, carbon dioxide, water, glucose, proteins, amino acids, fatty acids, sodium ion, calcium ion, chloride ion, ethanol, urea. So can you start thinking, which of this will be permeable and which of this will not be permeable? So think about oxygen. Will it be permeable? Think about molecular size. Think about the fact that it's a gas. It's a yes. And you already know why. It's a yes. Think about carbon dioxide, the same thing. It's a yes. Water, it's a yes. Glucose, think about glucose. We already established glucose. Think about its molecular size. Glucose, no. It will require specific transport proteins. What about proteins? Proteins, because sometimes we have small peptides in your small intestine, because they are broken down. Pepsin, chymotrypsin, trypsin, they break down the proteins into small peptides, and sometimes these small peptides are in the small intestine. and they need to cross. Will they be transported? Will they cross? Will they be permeable? It's a no. Yeah, so I'm going to allow you to take these other molecules, so think about them and do them as a classwork. You can discuss them with the chat bot, and if you have like difficulty in understanding the criteria, applying the criteria of charge, polarity, and molecular weight or size, then you can train yourself with concrete examples, or beginning with these examples using the course chat bot. Now, in this slide, we are going to be looking at how water crosses the membrane. So remember, we said that water, although slightly polar, it flows. It can cross the membrane, but quite slowly. But at the pace in which we need the exchange of water from the extracellular matrix into the cytosol, the rate is too slow. So because of the slight polarity of water, the rate at which it crosses the membrane is too slow. So we can't rely so much on simple diffusion. And that's why for water, we have special channels called the aquaporins. Aquaporins. So aqua for water, pour, like pour, P-O-R-S. Aquaporins for water. So these are special proteins. So when we're thinking about the special transport protein, we talked about transporters earlier on, and we differentiated transporters from enzymes, membrane transporters from membrane enzymes. Do you recall? We differentiated membrane receptors as well from membrane enzymes, and we differentiated membrane channels. So we have membrane channels, transporters, enzymes, receptors. So aquaporin is an example of a membrane channel, right? So if we look at, in the case of glucose, the transport protein that allows glucose to move in and out of the cell is also a specialized transporter. We'll come to it. It's called the GLUT transporter. So we'll come to that in a bit. That transporter is called GLUT. Okay? So GLUT protein. So we have the GLUT2, we have GLUT5, and so on and so forth. So depending on the tissue, GLUT5 is more in the kidney, and GLUT2 is found more in the small intestine. Right. So this is quite important. So, but the key idea here is that the protein that is needed to transport glucose in and out of the cell is called GLUT, and that for water is called aquaporins. So with this, we can summarize passive transport and the importance of passive transport. And well, I found this slide that can make you understand a bit better why it's important. So first of all, we talked about the exchange of gases. So you don't want any hindrance when it comes to gases. So gases need to move freely. The second is homeostasis, for balance, right? Balance of this compound. And so take, for example, water. You also want water to move a bit freely so that the cells do not get unnecessarily turgid, otherwise there can be plasmolysis and they can burst. So if water needs to move from a region of higher concentration to a region of lower concentration, well, that's osmosis. It needs to happen freely, and passive diffusion makes it possible. And the same idea for osmoregulation, water regulation and movement of water freely. Then we already established the need to conserve energy and adaptation to the environment, because the environment also, you can also have water coming in. It can be humid, if you think of plants especially. So there'll be need for that regulation and adaptation. And of course, it helps to just make metabolic processes easier. So in this slide, we're going to explain primary active transport, and we're going to look at the characteristics of primary active transport, and we're going to explore how it works in relation to secondary active transport. Okay, so one of the key characteristics of primary active transportation of molecules across the cell membrane is that it requires ATP. So this is a very key one, and we know what ATP is: adenosine triphosphate. So every primary active transport requires ATP. And how does this work? Usually, in a primary active transport, you have the transporter, right? But in this case, in a primary active transport, the transporter is also working or acting as an enzyme. So these are very critical examples, and I hope it doesn't cause any confusion. So it acts as an enzyme, but at the same time it's transporting. How does it work? In the cytosol, you remember that this is the extracellular side of the membrane, and this is the intracellular or cytosol. So in the cytosol, you have ATP, and usually ATP has 3 phosphates. So when we talk about phosphorylation reaction in the cytosol, what happens with this enzyme, a membrane enzyme, is that ATP releases 1 phosphate, and that phosphate is then used by the enzyme to catalyze this reaction, which I'm about to explain. So the phosphate binds to the enzyme in the membrane, and through that process a catalysis reaction occurs. And in this catalysis reaction, what happens is that as the phosphate binds, a conformational change is induced. A conformational change is induced in the enzyme, and as a result, this conformational change leads to the movement of sodium in the cytosol. So you can see all these balls, they are represented as sodium. So this conformational change leads to sodium, 3 molecules of sodium specifically, binding into the enzyme or binding to the enzyme. And what happens is that sodium, 3 molecules, is pumped out from the cytosol into the extracellular space. So I'm going to take it again. I'm explaining primary active transport. We already established that it requires energy, and because it requires energy in the form of ATP, in the process ATP is hydrolyzed, and the phosphate group from the ATP binds to the enzyme, and the enzyme this leads to a conformational change. The conformational change then leads to sodium being pumped from the cytosol into the extracellular space. And a very good example of this active transport is sodium potassium ATPase. And actually, sodium potassium ATPase is the enzyme which I have just explained here. So we probably already figured out that this enzyme, membrane enzyme, is sodium potassium ATPase. Okay, so that is the basic principle of primary active transportation. In this slide, I will be explaining secondary active transport. And in the explanation of secondary active transport, I'm going to be using an example of a transporter found in the enterocyte, and that transporter is called the SGLT1. And SGLT here stands for sodium glucose transporter, and it's a variant. There are many of them, so this is the first variant, and this SGLT1 is found in the enterocyte. You find it in the apical side of the enterocyte. And just to recall, if you're thinking about the enterocyte, you know we have the apical side and we have basolateral side. Okay, just for you to recall. So, but don't forget that it's in the membrane that you find this enzyme. So you can liken this membrane to, you know, locating it in the enterocyte. So that's just for you to understand a bit better. Okay, so how does the SGLT1 transporter, how does it work in the process of secondary active transport? So two things that you can see here, continuing from the previous slide, you have your sodium potassium ATPase, which is a membrane enzyme, and here you have your SGLT1. So in practice, in reality, the two of them work together. So you have to see them working together within the membrane. As you can see within the membrane, they're side by side in that sense. So we already explained how the sodium potassium ATPase works in active transport. So what we're explaining now is how it works, you know, with another transporter, SGLT1 transporter, in co-transport. And this co-transport is what we refer to as secondary active transport. So how is it secondary active transport? I'm going to be explaining that in a bit. So imagine that during active transport, the membrane enzyme, sodium potassium ATPase, has driven sodium ions from the cytosol, 3 of them, into the extracellular space. So you then have a lot of sodium in the extracellular space. So you have a lot of them. And when we talk about concentration gradient, just for you to have a better understanding, when we talk about concentration gradient, you have at the beginning, you had high concentration of sodium inside the cell and a low concentration of sodium outside the cell. And what happens afterwards? Against the concentration gradient, the sodium potassium ATPase pumps the high sodium and pumps it into the extracellular space against its concentration gradient. So it's like a pump. So you can imagine a pump working, so you can see this like a pump. So pump pumps from a high, it keeps pumping it until it becomes higher in the extracellular space. Okay, so this is actually needed to create what we call an electrochemical gradient. And you will see how this electrochemical gradient is used in sodium glucose transporter to bring in glucose from the lumen of the small intestine into the enterocyte. Okay, so moving very fast, you can already see that sodium has been pumped from the cytosol, and now you have more sodium in the extracellular space. So as a result of the action of the sodium potassium ATPase, you now have in the extracellular space, you have high sodium and you have low sodium because the pump is working, right? The sodium potassium ATPase is working, right? So why do you need this? The electrochemical gradient, don't forget. So, and then we enter directly into the SGLT1. So in the lumen, you have glucose in the lumen from your food. So all the glucose that has been digested, from your eba, from your fufu, from your polysaccharides, all the carbohydrates now, converted into glucose, waiting to be absorbed. So what simply happens is that because you have a high electrochemical gradient in the extracellular space, sodium, all the sodium that has been pumped in the extracellular space then flows through the SGLT transporter, this time not against their electrochemical gradient. It flows along the electrochemical gradient, and it doesn't need a transporter, so it just flows easily. But as sodium binds, glucose also binds, so the force that makes sodium bind, that causes a conformational change or opening of the transporter, also brings in glucose, and as a result, glucose comes into the enterocyte. So this is the reason why we call this, technically speaking, we call it co-transport, because it doesn't require exactly ATP, but the ATP that it uses is the ATP from the sodium potassium ATPase. So it's like a free ride of glucose into the cell, depending on the ATP already generated from the sodium potassium ATPase. That's the primary active transport, which creates an electrochemical gradient, and while the sodium naturally flows back into the cell, glucose then follows it. So this is the mechanism with which glucose is transported through secondary active transport. And we call it secondary active transport because it doesn't require ATP directly, unlike primary active transport that requires ATP directly.